Hi everyone, welcome back. This week we'll be discussing penicillin. Specifically, we'll be talking about what penicillin is, what conditions penicillin is used to treat. We'll be discussing the chemical structure as well as the mechanisms of action of penicillin in the body. And we'll also spend some time talking about how bacteria have evolved over time to respond to penicillin treatment. Penicillin belongs to a class of antibiotics known as the beta-lactams, and these medications are used to treat infections caused by susceptible gram-positive bacteria, typically in the Staphylococcus or Streptococcus family, although there are some others that it can treat as well. Penicillin is arguably one of the greatest medical advances, and it came about in the 20th century entirely by accident. It was discovered in 1928 when Sir Alexander Fleming, who was working on microbiology models at the time, realized that he had a species of mold growing on some of his plates. And on these plates, the bacterial colonies were killed in areas where the mold was growing. He understood at the time that this likely had clinical significance. However, he was unable to purify pure penicillin and ramp up commercial processes. So it was not until the 1930s and 40s, during World War II, that penicillin purification was made a priority. Penicillin was not made commercially available as an antibiotic until 1944, after its approval by the Food and Drug Administration and its adoption into the U.S. pharmacopoeia. The term penicillin now is inclusive of multiple members of this same beta-lactam family, all which differ from each other by an R group attached to a common amide structure. Penicillins inhibit bacterial wall synthesis, which is important, and is the basis of their bactericidal activity in treating infections. Now that we know what penicillin is, let's talk about penicillin's mechanisms of action. Specifically, penicillin inhibits the last step of bacterial wall synthesis in gram-positive bacteria. As we mentioned, these bacteria have a cell wall that contains peptidoglycan. Peptidoglycan reinforces the bacterial cell wall and allows the bacteria to withstand a significant amount of osmotic pressure in the body. Since humans only have cell membranes and not cell walls, penicillin is specifically targeted to bacteria that have the highest concentration of peptidoglycan in their cell walls. The way that it works is by binding to specific proteins called penicillin binding proteins within the cell wall and inhibiting an important enzyme called transpeptidase. Penicillin does this by mimicking a peptidoglycan molecule, and it forms an ester that binds the penicillin to the transpeptidase enzyme. The bulk of the penicillin molecule, which I'll show on the next slide, prevents nucleophilic attack at the ester carbonyl. This, in turn, prevents peptidoglycan linkage. Now, if the peptidoglycan molecules cannot be synthesized and linked, it leaves the cell walls permeable to water, which leads to apoptosis because the cells simply cannot withstand the osmotic pressure without that peptidoglycan linkage. As we mentioned, penicillin is a very large molecule, and you can see that as evidenced on the right-hand side of the screen. The basic structure is an R chain bound to a lactam ring that is fused to a 5-carbon ring. The beta-lactam ring is the key to penicillin's activity within the body. Specifically, the C-double-O bond ring in the lactam is electrophilic, which leads to ring strain. The ring strain makes it more prone to nucleophilic attack. The carboxylic acid group is also important because it binds to positively charged amino acids within the active site of the transpeptidase, which is the enzyme that we mentioned earlier that's important for peptidoglycan synthesis. and the lactam ring is a four-membered amide ring. We've talked about what penicillin is and how it works in the body. Let's talk about how bacteria have evolved over time to respond to penicillin and why this is so important. Resistance to antibiotics in the penicillin family is common, with as many as 50% of isolates from American patients with bacteremia or bacterial infections in the blood showing some level of penicillin resistance. Penicillin can be converted relatively easily to an inactive form, penicilloic acid, in environments that are highly acidic in the presence of water. Now, this is important because natural penicillins need to be given in either intravenous or injectable forms to bypass the digestive acids in the stomach that can inactivate them. Now, this is not a concern so much with synthetic penicillin formulations because this has been specifically addressed. Bacteria can evade penicillin either by antibiotic modification or physiological modifications of active sites. Active sites can be retained, but antibiotics themselves can be inactivated through molecular processes, or physiological conformations of the binding sites, such as penicillin binding sites, can be changed to prevent antibiotic binding. Some bacteria even produce enzymes, such as penicillinase or beta-lactamase. In bacteria that have mutations allowing penicillinase production, the penicillinase enzyme cleaves the amide linkage in the lactam ring that we talked about earlier, which inactivates the antibiotic. Some synthetic analogs of penicillin, such as methicillin, have been developed that are not easily inactivated by penicillinase. Beta-lactamase is another enzyme that's very important because it cleaves the lactam ring, which also inactivates the antibiotic. Here are the references for today's discussion, and I thank you for spending a few minutes with me to discuss the importance of penicillin and how it can be used in the body. Thank you.